Well, uh, thanks so much for giving me the opportunity to present some of the work from my research group. So this is sort of a topic that we've been working on for many years, and my lab is sort of a synthetic biology group, and we do a lot of microbial engineering. And been recently, over the last 3-4 years, been involved with Taka and collaboration with David Kaplan's group. But this is a project that's sort of slightly different, more related to precision fermentation. And we've been focusing on trying to understand how do we engineer microbial platforms for production of value-added chemical from waste materials. And one of the projects that we started is on trying to engineer a baker's yeast with expanded substrate utilization capabilities. All right. Yeah, so in general, my lab is a synthetic biology group, which means we focus a lot on upstream genetic modification of microbes for a lot of applications. And over the years, it has focused more on sustainability applications than biomedical applications. But the idea behind this particular project is that we want to be able to create a platform where we can take flexible inputs, run a microbial fermentation system for various lengths of time, preferably, extended periods, right, months at a time in continuous fermentation, and produce any desired product of interest. And one of the goals behind this particular project is to be able to take flexible inputs, have flexible runtimes, and a flexible output. So you should be able to switch between any input and any output at any time during the fermentation. And so the part I'll focus on right now is on taking a flexible feedstock system where you can have any kind of feedstock fed into your fermentation system and then have your microbe utilize it and make some sort of value-added product. But one of the problems that people have found is that while there are a lot of different microbes that can use different feedstocks, they're often highly specialized. You don't have one microbe that can utilize everything. You have some microbes that can utilize one subset, another set of microbes that can utilize a different subset. But can you create a system where you have a single microbe utilize a variety of different feedstocks? Now, there are two approaches people have taken in the literature in trying to develop these kinds of systems. One is sort of this seek and employ approach where you look at microbes from exotic environments that can utilize a variety of different feedstocks. So for example, here I've given, maybe like a termite gut where microbes in the termite gut might be able to use woody biomass really, really well, but they might not be so good at utilizing other things, so you might have to use a co-culture type system. The other approach is to take a microbe that is well characterized and can produce a lot of different value-added chemicals and proteins like baker's yeast, Saccharomyces cerevisiae, or E. coli, or other yeasts that are quite commonly used in precision fermentation, and engineer them to utilize a variety of non-native substrates. And sort of that's the approach that we've taken, and we describe this approach as synthetic heterotrophy, where we engineer an organism, in this case our work is focused on baker's yeast, to metabolize substrates that it normally did not evolve to metabolize. Now, the idea initially we thought would be straightforward, but over the past several decades, people have been trying to engineer baker's yeast to metabolize a variety of different substrates, and it's turned out to be more challenging than people initially thought. And largely, this is sort of because the idea of how do you engineer non-native substrate metabolism has been generally approached from sort of more of a metabolic engineering perspective, where you consider all the metabolic pathway in a microbe to be like pipes, and if you have the right pipe, you can metabolize the substrate. So if you just insert a pathway for a non-native substrate, in this case, let's call the substrate xylose, which is the second most abundant sugar in all of nature after glucose. It's found in inedible biomass, or inedible for humans anyway. Animals can utilize it quite well. If you just put in a pathway from a fungus that can utilize xylose, it turns out it's not sufficient to enable yeast to grow on this substrate or produce anything of interest from this substrate. And so, you can do a lot of optimization, balance metabolic fluxes to try and increase growth rates. And then if everything fails, you do what's called adaptive evolution, where you just culture your microbe in this substrate for extended periods of time, months, or I've seen papers where people have cultured yeast and xylose for up to a year to try and improve growth rates. And over that time, it accumulates a lot of different mutations that might enable it to utilize this particular substrate. Unfortunately, this is extremely, extremely time consuming. If you have to do a culture for 1 year for every substrate you're trying to metabolize, that is not a tractable approach for creating a flexible platform. So not only is it time consuming, especially this adaptive lab evolution step, just then trying to identify the mutations that led to the growth improvements and re-engineering them back is not trivial at all. And so, people have, what my conclusion from this is that evolution works really, really well. And so here's an example of, you can start with a small organism that is not so great at utilizing all substrates compared to one that is really good at utilizing substrates. So evolution has evolved organisms that are very good at consuming substrates. But evolution also leads to weird outcomes, right? So sometimes you get solutions that are not expected. Right? And so this is often what happens in long-term laboratory evolution. You get solutions that are not exactly what you're looking for, but are still evolutionarily fit. And so in general, microbes are kind of like kids. And these are pictures of my kids from several years ago where they're eating their favorite foods and they don't like to eat things that they don't like to eat. So over the past decade, we've focused on really trying to understand what limits the ability of baker's yeast to metabolize non-native substrates. We figured out that there's a synthetic gene circuit you can activate through this transcription factor called GAL4 that really changes everything about yeast metabolism, makes it very readily adaptable to non-native substrates. We've gotten it to metabolize 3, 4, 5 different substrates relatively quickly without any laboratory evolution, get wild-type-like growth rates very quickly. And so that's sort of where we are in this system. We're trying to create a yeast that is an omnivore, right, that can take any number of feedstocks in, metabolize them. And over the years, people have figured out how to make baker's yeast produce a lot of useful chemicals and proteins. And so that part is relatively straightforward to plug in. This is the part that we've been focusing our innovation on, is how do we enable yeast to metabolize a lot of these substrates. Anyway, so this project is sort of a relatively large effort for my group. And we have collaboration from the CELAG team as well. So Matt McNulty and Amin Nika, who are part of the TUCA, are part of this project as well. They've been helping a lot with the techno-economic analysis and life cycle assessment for this work. Some graduate students from TUCA, Lauren and Kirsten. And then there's a small company that we spun out that is also helping continue to develop this project. Todd was a former graduate student of mine, and Sam is an employee of Enrich Bio. I don't know how I did for time. I tried to speak. Yeah, all right.